It's a great honor today to welcome Professor Carmen Sandi as our first speaker to this symposium. Professor Carmen Sandi is a professor of neuroscience and director of the Laboratory of Behavioral Genetics at the Brain Mind Institute at the École Polytechnique Fédérale de Lausanne. Her work is combining approaches in rodents and humans to understand how stress affects brain function, behavior, and cognition. A special focus in her current work is placed on the role of mitochondrial function and the regulation of behavior by stress and anxiety by these organelles. Carmen Sandi has published over 220 articles in major international journals and contributed to many books. She also has many editorial commitments, as she was the founding editor-in-chief of Frontiers in Behavioral Neuroscience and is currently review editor for eNeuro. Carmen Sandi received many awards during her career, the latest being the Wonder Cloud in 2018 for her excellent contributions to stress research. Professor Sandi, thank you very much for accepting our invitation to this symposium, and we are all looking forward to hear your presentation named Brain Mitochondria and Metabolism on the links between stress, anxiety, and motivation.
Thank you very much, Adrien and Anaïs, and I would like to thank also the committee for inviting me to join this symposium. I think it's very impressive, the lineup of speakers that you've put together. I would like to ask you now, before I start, if you can hear me well. Yes, so everything is working well. Excellent. So yes, I think at the point I have to share my screen. I'm going to see if it's working now. Let's see here. Okay. So I'm also looking forward, by the way, to the discussion afterwards. I understand there will be some questions after this talk and then a general discussion after the morning sessions. And what I wanted to discuss this morning with you here is indeed the role that we are uncovering that mitochondria and also brain metabolism has in the different aspects maybe related to the production of behavior, but very particularly we think it's relevant for how stress, anxiety, and motivation interact. And I think this is going to get clearer throughout my talk. One of the main motivations, of course, that we have is to uncover novel mechanisms that we can target with different therapeutic approaches. Maybe some of them will become obvious to you towards the end of the talk. So when I talk about motivation, of course, motivation is a complex process, cognitive process that involves many different aspects. We are motivated to achieve incentives to obtain rewards. So we have motivation that it's based on effort towards achieving goals. And also there is motivation to avoid aversive situations. So this is mediated in the brain by similar, but also by some slightly different mechanisms. I'm not going to deal with these differences. I'm going to maybe share with you some of the reflections that we had in which aspects are important to achieve goals, either positive or negative. And now we have the program in the lab to understand the different aspects and the different also brain regions and cell types and connections that are mediating the different types, and also what's the role of mitochondrial metabolism there. But the thinking here, I think it's quite well reflected by these different pictures in which we see different situations of individuals that are trying indeed to achieve goals. For example, in this lower picture is achieving knowledge, going through content, something that we are very, I think, familiar with in our field. We need to put a lot of effort to finally get goals that are quite far. And I think here some of the qualities that are also depicted here, like having capacity to have resistance, to endure, to keep going are very important. And here, as we're going to see, there are important individual differences in how people can endure and persist. Also, there are other situations in which we are confronted with competitions, either of a more cognitive type or more physical type, and there again we need to have probably self-confidence. It's not only the physical or the cognitive IQ capacities, it's also other qualitative differences that are going to be critical to define which individuals thrive in achieving and getting these goals. So something that it's important, of course, in neuroscience, if we want to understand mechanisms, is to understand which circuits are mediating these different processes. But the thinking for this talk goes beyond that, if you want, in trying to be aware that we are very different in responding to different challenges, and also in being able to exert effort to acquire the different goals. And this is a challenge on itself for us to understand which could be key factors that determine that some individuals maybe have more active coping under challenges versus passive coping, etc. And if you realize in some of these pictures, or many of these pictures that I include here, or situations, we have situations of competition, because this is some of the aspects in which my lab has been focusing a lot, and that it's going to be the main focus of the talk, to illustrate the phenomenon, even though we are covering also the other situations, and we can talk a little bit about it a bit later. So, but the title indicates not only motivation, also how this is linked with stress and anxiety. And indeed, we over the years have identified anxiety, particularly trait anxiety, as an individual temperamental characteristic that defines a very important range of differences in susceptibility to stress, but also to exert different coping strategies under different challenges. And when we talk about trait anxiety, I want to make sure that we understand the same, we are on the same page. I'm not talking about anxiety disorders, I'm talking about the continuum of temperament that we all have in the population, and that is depicted here by a Gaussian curve, in which trait anxiety is going to define the different characteristics of individuals over time. They are quite constant in terms of emotional stability. So we know all that some individuals are very emotionally stable and quite cool, etc., very low in trait anxiety, and others are very afraid of many things, highly cautious, etc. So this is also perhaps a minority, but can represent a considerable percentage depending on the situations. And then most of us, in theory, would be in this kind of average. But of course, we have, again, a range of differences here. Of course, trait anxiety is one of the main traits related to neuroticism, that it's one of the 5 big factors of personality. So why trait anxiety in this context? Indeed, I was indicating that when we are exposed to challenges, stress, etc., there are important individual differences. Some individuals cope quite well, and some other individuals very quickly are more prone to start showing signs of different types of weaknesses. And if we define individuals as high or low anxious, according of different instruments, if we are talking about rodent research that we do in the lab, or human research that we also do in the lab, we use for rodents different tests which are quite well established and validated, like the open field, for example, and animals will be avoiding the center of the arenas if they are highly anxious, or the elevated plasmace, and they will be avoiding the open arms of these elevated plasmace if they are highly anxious. We define them very well, as you are going to see. And then we see that when we expose them, for example, to situations of chronic stress, the animals that are highly anxious are showing earlier signs of, let's say, depressive-like behavior, but also in the context of this talk, all alterations in the capacity to exert effort to achieve rewards. And also when we define individuals, human individuals with the state trait anxiety inventory, state T, that it's quite used in the neuroscientific literature, we also see in the literature that they are more vulnerable to stress. So, as I said, I'm going to tell you quite a lot about the work that we've been doing in the field of social competition, as we have intrinsic motivation to thrive, to achieve dominance by evolutionary means. We've been adapted throughout evolution. As you can see here, there are many different animal orders and species in which the individuals are always motivated to win fights. Many times dominance is achieved through fights in order to gain rewards and also be access to more resources. And what, of course, if one goes to the evolutionary biology literature, what you encounter is that the outcome of social competition is highly determined by physical traits, by the strength, the size of the animals, etc. This has, of course, not been so interesting for us. What we are particularly interested is whenever individuals have similar physical capacities and they are competing for dominance, which individual would have the mental strength, if you want, to be able to finally achieve the dominance rank, the dominant rank, versus the opposite, which individual would be subordinate eventually because of not striving to win the competition. So here in this slide, we have the formulated question: which factors affect the outcome of a social competition when the individuals are equivalent for body size, weight, and life history? And that's something that, of course, it's not so easy sometimes to tackle in humans, that we can equal all the different characteristics, but still we can look throughout different traits and see whether they vary together. And as you can see here, this is results that are based in specific questionnaires. We have that those individuals that vary on trait anxiety, the ones that have higher trait anxiety, these are the ones that have lower dominance motivation. And I think more strikingly, those that have high anxiety, that have less competitiveness, they are willing less to compete. And also they have a lower self-perceived social status. And this is with questionnaires, but we found very interesting results also when we did behavioral economic experiments in which individuals were performing economic games either under basal conditions or following exposure to stress. This was the trial social stress for groups, in which they were given the possibility to earn money either by playing a lottery or competing with another individual that they had been paired on the basis of their IQ that they had determined, or we had determined through a previous questionnaire or test that we gave them 1 week earlier. I'm not going to go into details, otherwise I wouldn't have the time. But what was very interesting that we found here is that those individuals that are low anxious are already slightly more self-confident in competitive settings than the high anxious under basal conditions, but under stress, they become, the low anxious, very self-confident, really overconfident, and the high anxious do the opposite. They become very underconfident. So as you can see here, what the stress is doing is revealing something that is probably a latent characteristic of these individuals, that high and low anxious have different predispositions to believe in themselves in the competitive setting. And that's something that we see in huge differences. And one could imagine that indeed this is something that happens in real life. And this is something that we certainly validated for animals. We can now indeed make sure that they have all equal characteristics. These are 2 male rats, same age, same size, same body weight, etc. Same husbandry, all the same conditions. The only difference that they are going to have is that we characterize them for trait anxiety in this test that I indicated before, the elevated plasmase, also the open field, and in this case here as well, the LiDAR box. And we separate now the animals that are highly anxious. These are the ones that they never dare to go into the open arms, but also they don't dare to go into the light compartment of the LiDAR box. And we call them high anxious. Then we have some of them that indeed they are intermediate anxious, and the others are very low anxious. And what we want to make sure now is that we have two different phenotypes, and we started asking high and low anxious animals to compete for a territory or for different resources. And what we consistently see is that the high anxious animals are the ones that almost systematically lose the competition. Not all of them, I have to say here that around 20-25% of the high anxious animals are very competitive or capable to win the competition, but we have a huge capacity to predict the outcome of this competition just by defining the animals according to this social hierarchy, to this trait anxiety. So the situation we have now is that indeed we can have a trait that a priori and without exposing the animals to any type of direct fight is indicating to us which animals would have the capacity to win the competition, to keep going, to endure, etc. And they're also under chronic stress, to be less affected, to fight more when they are confronted with challenges. And this would be more the low anxious versus the high anxious would be the ones that are going to be much more affected, retract quickly, etc. So this gives us now a very important tool to be able, or very valuable tool to be able to interrogate mechanisms that would be different in the two different types of animals, so that we can afterwards probe them and see if they are key in defining these different strategies, particularly under stress and in relation to trait anxiety, as I indicate here. So what we had to do originally was to understand which brain circuits and also brain regions and also brain circuits would be key for us to start looking into. And what we did originally was to confront high and low anxious animals to fight together, and afterwards look for brain activity measured by immediate early gene. We use different type of probes, and this is the results for CIF, in which you can see that when we compare with basal levels and look at many different brain regions, we have that activation in several brain regions, in particularly in the prefrontal cortex that we know from the literature, and very nice work by Hailan Hu and others that shows that the prefrontal cortex indeed gets engaged when animals are fighting to establish a social hierarchy. But something that was quite remarkable for us is the huge activation that we find in the nucleus accumbens. Indeed shows this activation when the animals are fighting. And we also did inactivation experiments in which one of the animals had inactivated the nucleus accumbens before social competition. In this case, both animals were intermediate anxious. And what we could see is that the one that had an inactivation of the nucleus accumbens lost the social competition. So a few words about the nucleus accumbens for those that may not be so familiar. Nucleus accumbens is part of the ventral striatum, and it's of course very well known as the recipient brain region of dopamine from the ventral tegmental area, and also receives excitatory glutamatergic projections from many different brain regions, but particularly medial prefrontal cortex, basolateral amygdala, ventral hippocampus. Also it receives inhibitory inputs from other brain regions. We don't need now to have the whole picture, and I will be talking later about outputs from this region. What is critical now to summarize in this context is that over the years there is a huge literature that's been developed showing that the nucleus accumbens is very important for reward learning, also for many different types of motivation. And here we have to also indicate that we have a core and shell different subregions, but there will be time maybe during the questions to go through those aspects. Something very interesting is that the nucleus accumbens is also being implicated in the vigor of responses, the strength that the individuals are putting in exerting different types of responses, also in the capacity to exert effort to achieve different types of rewards. We see in human studies that the nucleus accumbens pops up as activated in many of these studies in which they are looking for social hierarchies, whether the individuals would react to the other one being dominant or subordinate, etc. So it's critical there, and also in studies of incentive motivation, it's typically engaged. And also very relevant in this context of the talk is that the nucleus accumbens is highly implicated in the development of different symptoms related to depression, particularly when individuals are chronically stressed and they develop sociability deficits, anhedonia, also anergia, something that we see in many depressive patients, this lack of energy to be engaged in activities, and also showing low motivation, and also learned helplessness, and that I will be talking a little bit about in a few minutes. So indeed, we could see that it made sense to focus in the nucleus accumbens to get started in this type of projects, and to ask which mechanisms could define that high and low anxious animals are going to be different in exerting this different type of effort-related behaviors. But of course, when we talk about nucleus accumbens, we have a diversity of cell types, and in order to look for mechanisms, it's important to start understanding which cell types could be particularly involved. And here in this diagram, we see that there are the dopamine 1 receptor containing medium spiny neurons, and also the dopamine 2 receptor containing neurons. As I told you before, nucleus accumbens receives dopaminergic projections from the ventral tegmental area. So these two different cell types constitute around 95% of the cells in the nucleus accumbens. There are other important cell types as well, like cholinergic interneurons that are also very engaged or implicated in the release of dopamine, also GABAergic interneurons, etc. Basically, what we started asking in this work, because of the prominence of the medium spiny neurons that are also projection GABAergic neurons, is whether we can identify in these different cell types which ones are particularly activated when the animals are competing. We also do it for other types of motivated behaviors, but here we are concentrating now in the social competition. And what we found is that indeed we already knew that there was going to be an engagement of this cell, of this brain region. And in terms of cell type, we found that the neurons that contain substance P are the ones that also normally express the dopamine 1 receptor, are the ones that show a particularly increase in activity when we compare the different basal conditions with social competition, and not so much other cell types. And then when we compare high anxious and low anxious animals, you can see that it's particularly the low anxious that are eventually going to also win the social competition, that show a higher activation of these D1 medium spiny neurons. We also interrogated whether they would be causally implicated, mechanistically implicated by microinjecting in the nucleus accumbens shell an antagonist of the D1 receptors. And then we could see that they play really a critical role in the outcome of a social hierarchy, indicating that this is an important cell type in this context. So of course, we have different individuals that are engaging a particular neuronal type in different ways in the medium spiny neurons that can maybe indicate something that it's relevant in comparison between the high and the low anxious individuals. So we started looking at different possible differences in neuronal structure and function. And as you can see here, when we do Golgi experiments in medium spiny neurons, we see that high anxious individuals show a much poorer dendritic atrophy, that this is now quantified here in the Scholl analysis. And also they have a lower density of spine, so lower spine density, that correlates with trait anxiety with the time that the animals spend in the open arms, meaning that those that are more anxious, they have less dendritic complexity, as we can see here, but also across the continuum, and also lower dendritic spines. Of course, this was in medium spiny neurons. We want to know if D1 containing neurons, the one receptor, would also be different and particularly different. And this is something that we did here by filling cells that were recorded for electrophysiology with biosighting. And you could see here that, again, the high anxious animals have a lower dendritic complexity in this case. And this was then verified also by electrophysiology, in which what Simone Story found is that the high anxious animals show lower frequency of excitatory inputs onto the medium spiny neurons in these nucleus accumbens, and this was particularly, as I said, in the D1 containing neurons. So what mechanisms could be responsible for these differences between high and low structure, but what could be mediating these differences? Many years ago, we decided to do a microarrays to see if we gather any hints of where to go to try to understand these differences. And what turned out from those experiments is that many genes that were or are different, differentially expressed between high and low anxious animals were enriched particularly, and they were genes related to mitochondrial function and metabolism. So we decided to start looking into these aspects, but of course, before we had to make sure that it made sense to look at mitochondria and metabolism in this context. I'm going to make a little parenthesis to motivate the importance of looking at mitochondria and how this field has evolved, let's say, in the past 5 to 10 years. First of all, I think many of you are very aware of the importance of many mitochondria for many different functions. Particularly, it's very well known as the powerhouse of the cells, the production of ATP, and this is done through many different processes that are depicted here, like the TCA cycle, fatty acid oxidation, and amino acid metabolism, and also the production of oxygen, reactive oxygen species, ROS, that it's a byproduct of oxidative phosphorylation in mitochondria, but mitochondria are also very important for regulating calcium flux, and they have roles in apoptosis in many different processes. So I think the importance of mitochondria for the cell has always been clear. Maybe the importance of mitochondria in neuroscience is only becoming more and more clear in recent years. Normally when people, or when including ourselves, did this type of microarrays in the past and found that mitochondrial genes were enriched in the differences, we many times thought this could be due to housekeeping differences, something not so important, metabolic, not so important, maybe not so specific. And I think the field is changing a lot at the moment and recognizing that they play a very central role in many of these processes. Something that was particularly relevant for us to really decide to focus on mitochondria is that it's been also found that glucocorticoids, when they are activating the glucocorticoid receptor, they can also affect mitochondrial function. They can also translocate to mitochondria and affect different functions, including mitochondrial DNA transcription, but also glucocorticoids, as we know, they are transcription factors and also interact with transcription factors. And they can also modulate the expression of many different genes related to mitochondrial composition. This would be in the nucleus in this case. So of course, we know that glucocorticoids are main hormones of stress and very important also in the context of anxiety. And that's why it was important to look as well. So very quickly here, over the years, I think the field is exploding in showing the importance of mitochondria for brain function and also psychiatric disorders. And particularly in this context of psychiatric disorders, the thinking goes that the brain is a small organ in comparison with the rest of the body, but it consumes a lot of energy. And if mitochondria is compromised, it makes sense that the brain is going to be one of the first organs that it's going to show alterations in function. Also, there is now emerging and consistent evidence that mitochondrial DNA defects, either genetically or acquired, both in humans and animals. There is a lot of clinical work, also leads to alterations in different types of behaviors, and also very particularly individuals show some psychiatric comorbidities and alterations in anxiety, depression when they have mitochondrial disorders. Also, I think this is something we are not so aware, but mitochondria are very critical for the production of many hormones, including glucocorticoids, and also for the metabolism of catecholamines that are important brain neurotransmitters, and also critical for the stress response. And the idea here would be that indeed under stress, we need to exert, to adapt, and for this we need to exert a lot of brain energy, and that's why mitochondria would be critical in this different context. Work from our lab, and this was a paper that was finally accepted yesterday, has also shown that when individuals are exposed to chronic stress, these were mice, but we also compared with data in humans, mitochondria DNA encoded genes of different types of complexes get affected very particularly. This was one of the most prominent results. And what was, I think, quite intriguing is that if we look at whether individuals that are more vulnerable to the effects of stress are more or less affected in these changes, we find that indeed those that are more vulnerable are systematically showing, in this case, higher expression of mitochondria DNA encoded genes. So this was the if you want, a parenthesis regarding the importance of mitochondria. In our work, we ask if, when we look at the nucleus accumbens, we would find differences in mitochondria in different types of aspects. And of course, we started with function, with the gold standard in this case is to look for mitochondrial respiration, and this was done with the technique that it's a high resolution mitochondrial respiration analysis. That's with the Ouroboros method, in which we can measure how much oxygen consumption or mitochondrial respiration we have depending on different complexes of the ETC of the mitochondrial chain. And what we've been observing systematically is that high anxious animals have lower maximal respiratory capacity than the low anxious animals. It's stable over time, and it's in neurons and not in glia. And we also find that it's not only activity or function, if you want, we also found differences in the expression of the protein for these complexes. When we measure ATP content also in these in nucleus accumbens, the high anxious animals have lower ATP and also high levels of ROS, indicating that indeed mitochondria seems not to be working so well. And when we probe if the function of mitochondria is important for the outcome of the social hierarchy, we find that indeed it is. So we have also causality when we manipulate specifically complex 1 and complex 2 activity in the nucleus accumbens of these animals for the outcome of the social competition and for other motivated behaviors. So why would mitochondrial function be important for these neurons? So here is electrophysiological recordings in which you can see that manipulating complex 1, inhibiting a little bit, so too much could be toxic, reducing the activity of complex 1, we find a reduction on the firing frequency in the nucleus accumbens medium spiny neurons, and not so much in other types of neurons, as you can see here in cholinergic neurons. And when we play with the levels of ATP, we see that particularly, again, medium spiny neurons are very sensitive to the levels of ATP. And this is now for the D1 containing medium spiny neurons, even though sometimes there is a lot of co-expression of D1 and D2, and it's not excluded that, of course, D2 containing neurons can also be affected. So we have now probed differences in mitochondrial function. How about mitochondrial structure? If we look at mitochondrial structure, can we find differences between high and low anxious animals? And as you can see here, we do. We can measure many different parameters. We have here the mitochondria that it's filled with cyan color. And then you have in low anxious animals that it's smaller. In the high anxious animals, it's bigger. Sometimes we find it also more globular. So when we measure parameter like mitochondria coverage on the image, it covers much more. But this doesn't come with a bigger or larger number of mitochondria. The density is the same. Something interesting that you see here is that indeed mitochondria has interactions with other organelles and very particularly with the endoplasmic reticulum. And this partnership is something that we quantified here, in which what we could find is that high anxious animals have lower contacts between the mitochondria and the endoplasmic reticulum, even though they have more contact between the same organelles. And this, we thought it's a very relevant observation to follow up, because the interaction between mitochondria and the endoplasmic reticulum is key for many different factors. Also, these differences in mitochondrial shape are indicative that maybe differences in mitochondrial dynamics could be affected. And we looked into genes that are important for all these two aspects, for mitochondrial dynamics, all the ones you have here, mitofusins 1 and 2, DRP, OPA1, and also for the interaction between mitochondria and the plastic reticulum. This is particularly mitofusin 2. And what we found is that indeed high anxious animals have lower levels of mitofusin 2. As you can see in these images, it's particularly in the nucleus accumbens, we don't find the same in the amygdala. So this was very encouraging, because indeed the structure is telling us that this could be a key aspect, that it's causally implicated in the differences that we observe. Mitofusin 2, as I indicated, is important for fusion, also for this tethering, also for motility. So it's a possible mechanism also to account for differences in the structure of the cells. If mitochondria cannot get to terminal parts of the cells, that could be one reason. So we decided to probe causality by injecting animals with a virus, AAV virus, that's going now to overexpress mitofusin 2 in the nucleus accumbens of high anxious rats that normally have lower levels. And then we looked at all these parameters. And since I think time is running, I will try to go a bit quick here, because everything worked. Everything that I told you until now that was different, or maybe that high anxious animals had a deficit. Like, for example, in mitofusin 2 RNA, of course, we recover this, but also in mitochondrial respiration, we recover it in the high anxious animals by overexpressing mitofusin 2. Also, we looked at mitochondrial structure, and we did a 3D reconstruction in order to have a better idea, because mitochondria are not 2D, they are really globular, and they cover many different fields. And you can see it here, I think, by naked eye, if you want, that there are differences. When we quantify the differences, they come up as quite striking. Indeed, overexpressing mitofusin 2 in the high anxious animals completely recovers the differences between high anxious and low anxious, like the surface, the volume becomes more normal, they are less globular, and also the length, etc. And the number continues being the same. Also, when we look at the neuronal structure, you can see that it's quite impressive. We completely revert the atrophy of these dendrites. The high anxious animals, medium spiny neurons structure becomes very similar to the low anxious animals. In terms of connectivity also, excitatory inputs are recovered. And in terms of behavior, now high anxious animals that overexpress mitofusin 2 don't lose against the low anxious animals. They, in fact, win the social competition. So by targeting mitochondria in these cell types, we completely revert the phenotype. So now we are trying to understand better how does this account for the circuitry? Now we know that D1 containing medium-spiny neurons, mitochondria, seems to be critical for these different type of coping behaviors, vulnerability to stress, if you want, capacity to win. And now we are looking at downstream mechanisms. I'm not going to go into details. This is a lot of ongoing work, but so that you know, we are now using mice, with viruses also that allow us to look at specific outcomes of this circuitry. So to summarize this big part, we could identify indeed that nucleus accumbens is important, and even more, the mitofusin 2 in medium spiny neurons plays a role in determining mitochondrial structure and function, but also neuronal structure and function, and the outcomes that are mediated by this circuitry in terms of stress coping. I'm going to now very quickly, I think I have 6 more minutes. Is that the case? Yes, I think so. With what type of behaviors are affected by mitochondrial function? I've been claiming that it's not only nucleus, it's not only social hierarchies or the outcome of a social competition. Indeed, we also see that active coping under adversity or floating in the force-win test is affected by this function. And also we are looking into the operant conditioning, the capacity to exert effort to obtain rewards, and there are differences in the breakpoint in operant conditioning. This is just to give you the feeling that indeed it's not only the social competition that it's affected. If we have animals in which we reduce the activity of complex 1 of the mitochondria, etc., in the nucleus accumbens, and then we put them to cope with adversity, let's say in this beaker that is full of water. And as you know, normally what we measure here is floating behavior that is normally depicting of a passive strategy under stress. We see that those animals with the inhibition of complex 1 have a much higher floating behavior, which is not because they are affected in the locomotor behavior at all. It's really the different strategy that they are impinging in this type of behavior. So why would it be that there are these differences? And one could imagine that maybe genetics could play a role if there is variation in the expression of these genes. That's something we are looking into now in this outbred Wistar rats. But of course, we know that early life stress can program many different functions, and we've been looking into whether or not this could be relevant for this type of questions, and asking specifically whether early life stress could affect the later mitochondrial function, and how does that work? And indeed, we used a model that we developed in the lab in rats, and now we translated into mice that we call it peripubertal stress. And it's based on exposing animals to fearful situations, to challenges, to stress at childhood and puberty. And it leads to many different behavioral outcomes, including a reduction in sociability, that's mediated, as I indicated, by different regions, but also including the nucleus accumbens. And what we observe also is that 2-3 months after the animals were exposed to this stress, when we look at mitochondrial respiration in the nucleus accumbens, there is a reduction in those animals that were stressed. And when we try to look at mechanisms, this project, I'm going to summarize it in 1 or 2 minutes, we, among other differences, we see that NAD+, which is a very important cofactor for many different energy processes, including CO2-1 mediated functions, is reduced. So a critical energy-related component in the nucleus accumbens is reduced many months after they were exposed to peripubertal stress. And what we are finding here is that there might be, in fact, a link between brain fat and some of the signaling that goes from some adipokines. In particular, we identify ENAMPT could be signaling the brain and affecting the adaptation of different functions and also behaviors. And this is one way in looking at how all this could be happening from periphery to brain. It's probably a fat to brain excess. And we are doing some work in humans also to see the translational impact of all this research. We start by doing a magnetic resonance spectroscopy in mice, so that we can indeed go from the differences at the molecular structural level, mitochondria, into metabolites by using this spectroscopy imaging. And what we could do with this type of approach was to identify different metabolites. I'm not going to go into them because of, on the sake of time, that are different between dominant and subordinate animals, and also that they change in different ways when animals are exposed to stress, and the animals that are vulnerable or not, they show differences. One metabolite that you can see here is highly different between high and dominant and subordinate. This would be subordinate is taurine, that have lower levels in the nucleus accumbens, and this is reverted by or affected by stress. And indeed, we see that both high anxiety and chronic stress it goes together with low levels of taurine in mice. Taurine is important for many different functions, including the functioning of mitochondria. And when we look in humans, also with spectroscopy approaches in a 7 Tesla, we found that taurine levels in the nucleus accumbens variate also with trait anxiety, so that those animals, those people that are more anxious, they have also lower levels of taurine as animals. So that's quite similar, and that's the way and the approach that we are doing. We've been trying different treatments. Of course, many of them not drugs like diazepam that works quite well in affecting mitochondrial function and also all these different functions, but also others that are more related to nutraceuticals or nutritional approaches. And as you can see here, I listed the different type of papers that we have published. I'm going to jump the first one, which was based on the medium chain triglycerides, which I think gave magical results. It reduced anxiety. And a more recent work in which what we've been doing is to identify individuals that are vulnerable to chronic stress, and then doing spectroscopy, in which we see that indeed there are differences in the metabolites that are expressed. And then we gave a treatment with L-acetylcarnitine that also targets mitochondria and boosts mitochondrial functions, particularly the import of fats. And we, as you can see here, we have a complete recovery of the negative effects of stress in or chronic stress in taurine and glutamine, for example. So the idea is that indeed by these nutritional approaches, we can affect brain, mitochondrial function and metabolism, and also revert the behaviors. And something else that it's very relevant in this context is probably treating the glutathione pathway, and that's something that it's reviewed in this review here of the literature. I don't want to take more time because I think it would be fun to have questions. Just finalizing without summarizing more that indeed this is our working hypothesis. A role for mitochondrial metabolism is key for neural coding for stress coping and motivated behaviors, and it's modulated by anxiety. And I would like to thank the wonderful lab and collaborators that we have. I didn't have much time, but I acknowledge the key contributors to all this type of work, and you have here their names. And I thank you very much for your attention.
Thank you, Professor Sandi, for the very nice talk. We now have time for 7 minutes of questions. Since we didn't receive any questions yet in the Q&A chat, maybe we can start with a very general one. We were wondering if the results you showed also included data from females.
Yeah, no, I didn't show yet data in females, but this is a very interesting question that we are addressing now specifically, because of course we started doing social competition and it's more evident, if you want, in males. It's easier to establish, and we've been developing all this in males. Now we indeed have the question in females to see if the phenomenon is similar, but also it will be very important, I think, and interesting to compare to compare the two genders or sex for these different aspects, because we know, for for example, that when we look overall to the population, females, both, but particularly humans, are more anxious on average than males, and then also less competitive, as you could see in some of the questionnaires that we show. Not, of course, not all the females, but it will be interesting to ask these questions. Yes, definitely.
Thank you. Erocastren is asking, do this nutritional treatment of peripubertal stress regulate Mitofusin too?
Thank you very much, Ero. Nice to talk to you now. It's something that we are now measuring. Of course, we have very interesting data in where we really looked into it now in details in the latest or the last model that I show you based on peripubertal stress, in which indeed just exposing animals to stress at childhood and puberty, then we look at adulthood, gives reductions in Mitofusin 2 that are programmed 2-3 months later on. And then we gave a treatment which is based on this function of NAD pathway. It's NAMPT and NMN that was given in the drinking bottle, and also by overexpressing NAMPT in the adipose tissue. And indeed, we could recover the Mitofusin 2 levels. So now this opens, of course, questions as to the nutritional approaches that are more linked to the Mitofusin 2. So something obvious that we are now putting in place is looking at taurine, that it's very important for this pathway as well, related to the dynamics of mitochondria. Thank you.
We have another question. Would you link the altered function of mitochondria in high anxiety animals to changes in calcium homeostasis, or rather to changed energy status of the neuron?
What I didn't understand well the question. Which homeostasis?
This person would like to know if the altered function you observed in mitochondria in high anxiety animals were due to changes in calcium homeostasis or general changes in this energy status, but specifically in neurons.
Yes, it's very relevant question. We are now addressing it with mini endoscopes, and it's something that is ongoing work. So of course, calcium is going to be affected, but what's first, that's something that is not yet resolved in the lab. If we have a direct effect, that could be the case, because we know that glucocorticoids exert a transcriptional control on the levels of Mitofusin 2, so we wouldn't need to go through differences in calcium due to differences in the activity of the circuit. But I think, of course, one cannot exclude it, and it's very normal that indeed mitochondria responds to the activity and changes in calcium. I think, there are a lot of very interesting questions also with these differences in the interactions between mitochondria and the ER, being these interactions so critical also for calcium processing and buffering. It's going to be important to tackle this specifically, and that's something that one can do these days by using probes that change the length or the distance of these interactions between mitochondria and the ER, but this is something that we'll have to do in mice. We have now a lot of work ongoing in mice at the moment.
Okay, thank you a lot. Could you specify which glial cells you looked at for the ETC respiration experiments?
Yes, in that case, we did astrocytes, and now with the Cartana approach, we could really see also with the project of early life stress that we cannot exclude that there are differences in glial cells. When we look in particular, we are focusing in the first step in astrocytes. We now think they might be quite relevant, and maybe not for differences in the respiration, but differences in the structure that we also see, or this kind of partnership or interactions between mitochondria and the plastic reticulum. We see that astrocytes also show very interesting differences that we are now, right now, there is a postdoc in the lab tackling this. So I said for mitochondrial respiration, we didn't see differences, but we don't exclude that this is a contributing factor. In fact, it's a bit the opposite, because the results that we have from metabolism with the spectroscopy systematically show differences in metabolites that rely from glial function, like lactate, for example, even taurine eventually, and also other differences. Alanine, we have also differences. So it's not, we don't exclude the importance of glial cells, and we are looking at astrocytes.
Okay, thank you very much for your answers. I think now it's time for a short break, a 5-minute break before our next speaker, and we are very excited to hear you during the panel discussion that will be at 12:10 Helsinki time. So we are all looking forward to this. Thank you very much for being
Carmen.
there. And
thank you. See you soon.
See you.
Thank you.